So again, welcome to the third annual day-long course on advances in brain tumor treatment and management. And this is a heavily sort of translational research-oriented course. Most of the courses at Seattle Science Foundation are very practical, hands-on with cadavers, et cetera. But in the brain tumor field, it's always been my thinking that the critical advances that we're going to need for this field are not going to come from, not necessarily from better surgical techniques or better radiation techniques, even though those have a significant impact. And I'll just kind of walk you through. I like to start with this slide every year to show what we're dealing with and where we're coming from. Ooh, forgot that this was that fancy. So over 50 years ago, before serious surgical resections were done for glioblastoma and radiation, the median survival for this disease was somewhere in the range of 3 months. It was considered pretty much a very dire prognosis, and patients were not really treated aggressively. With the advent of more aggressive surgeries, it actually doubled or over doubled the survival to median survival of about 6 months or 9 months. With the advent of radiation treatment plus surgery, the survival was again doubled to about a year. And then typical chemotherapeutic drugs slowly advanced, but not significantly. So basically surgery, radiation, and aggressive surgery and radiation were the modalities. And then I guess you could call it a relative breakthrough occurred in 2005 when 2 publications came out showing that temozolomide could improve survival for patients from a median of about 14 months out to a median of about 17 or something like that. And it was a big deal, although in my mind I felt like, that's not that great. And it turned out that that was primarily due to just a subset of 1/3 of patients who happened to have a methylated promoter for a gene that essentially inactivates the chemotherapeutic drug. So 2/3 of patients were not really benefiting from that, and that's still the status today. Along the way, more aggressive approaches to surgery have been utilized, and including some techniques that I know Dr. Berger and I have used, using fluorescent markers to try to get every bit of tumor during surgery. And one of the leaders in that area has been the group Tumor in Germany. And a meta-analysis of all the aggressive surgical approaches for glioblastoma does indicate that more aggressive surgery, if possible, does improve survival, but still we're not talking about a major breakthrough. We're talking about a few months, but every month counts. But what we're here to talk about today is the out-of-the-box ways that we're going to have to move this beyond incremental months of 16 to 18 to 20 to 24 months, whatever, and to try to cure the disease. And I think it's something that we're thinking about and talking about. The buzzword for a while now has been personalized medicine, using genomics approaches, and it has been wonderful for certain specific disease entities. Sometimes fusion proteins and mutations can be identified, but for glioblastoma in general, it's not been as easy of a approach as we had hoped. But there's still a huge role and rationale for looking into these approaches, I think, based on what we're learning from other things about these tumors. I think personally that 2 of the biggest breakthroughs or scientific areas of understanding that will lead us to improve survival are understanding the role of the so-called stem-like cells in glioblastoma. And we've got one of the discoverers of this cell, Dr. Dirks, in the back here. And he continues to elucidate the role of these cells, and as we can further understand this subset of tumor cells, which seem to be responsible often for the recurrent disease, I think we will have a good grip on how to deal with the disease that kills people, which is the recurrent disease. And at the same time, immunotherapy has expanded throughout all cancer treatments. We have multiple trials even here using sort of generic immunotherapy approaches, checkpoint inhibitors. But I think the end run will be when we get specific targeted therapies for glioblastoma. So I'm not going to go through these slides, but this just reinforces the fact that the glioma stem cell is a complicated entity, and there are all kinds of pathways that need to be further understood, but hopefully we can hone in on targeting these cells. And also, this slide is an example of a sort of current understanding of all the multiple factors involved in attacking glioblastoma cells, which seem to put out this yellow thing here. I think is kind of a haze of a force field against the immune system. And all these molecules like PD-L1 and other entities facilitate the immunosuppressive nature of this tumor. And these have to be overcome to allow an immunotherapy approach to function.